Prior to the Devonian period, there were no vertebrates living on land, but by the end of the Devonian period, amphibians had evolved from sarcopterygian fish. By the following period, the Carboniferous period, some of these were recognized as the earliest reptiles. It would take millions and millions of years for the reptile lineages to diversify. And so, things like dinosaurs, crocodiles, snakes, lizards, they were, tens and tens of millions of years in the future. Amphibians were quite successful for a while. And it should be noted that a number of them made adaptations to life on land. And so it isn't as if we have two separate categories of amphibians and reptiles. Many of these fossil groups were then in between. And so we could even argue as to whether we need new terms to describe amphibians, which are now adapting to land. So when we look at this animal here, look at the long ribs, the stout limbs, clearly becoming better adapted for life on land. One could point to very gradual changes. So in the earliest amphibians, the humerus was L-shaped, and the very earliest ones hadn't even settled on 5 being the appropriate number of digits. So that Acanthostega had 8 fingers and toes. Ichthyostega had 7, Tularpaton had 6. But eventually this would then be reduced to 5, which then became the standard for the amniotes. And they gradually got better at life on land. For example, if you're going to live on land, your legs have to support your body's weight. And so there are specializations of the ribs in the hip region. And the vertebrae can be modified to form what's called a sacrum. And so the formation of a sacrum, these vertebrae which were specialized with sacral ribs, which were specialized to support body's weight on land, this did not appear all of a sudden and developed in a series of stages. The earliest reptiles still had many features of amphibians and then even fish. So dermal scales, the type of scales that we find in fish, not reptilian scales, are still known on the underside of amphibians and then even reptiles. If we were to consider the first amphibians, they had a hole at the top of the head for light to enter and strike the pineal gland, which was photosensitive. This continues through the early reptiles. In fact, even in our lineage, through many synapsid reptiles as well. So the first reptiles had this amphibian feature. The first reptiles still had these large teeth on the roof of the mouth. And so the amphibians, the sarcopterygian fish before them, did have teeth along the margins, but they had these prominent fangs on the roofs of their mouths. The first reptiles would still have this feature. If you were to look at, say, the skulls, the bodies of many of these amphibians, such as a group called the anthracosaurs, they became better adapted to life on land, with more elongated humerus bones, with formations of the sacrum, stouter limbs adapted for supporting the body's weight on land. If you look at this hip and this anthracosaur, a greater area for muscle attachment than in earlier amphibians. If you were to look, say, at this pink bone, the stapes in the back of the skull, notice that it was becoming smaller, no longer used as much as a structural element for support, but rather this small bone became the first of the middle ear bones. The stapes, mammals would add 2 more. And so if you were to consider the amphibians, they gradually, or there were a number of diverse lineages, some gradually adapted to become more efficient at living on land, supporting their body's weight, etc. And so then at some point in the Carboniferous period, there were reptiles. Now, the earliest reptiles, even though we know reptiles, would eventually form these huge dinosaurs and crocodiles, enormous size, the earliest ones were quite small, all right? And that makes sense given what I'll say about reproduction presently. So if you were to say, all right, what were the first reptiles look like? Well, they were, very small insect eaters in all probability. So while reptiles did appear in the early Carboniferous period, they were not the reptiles of today. And so we could point to a large number of features which these first reptiles had, which were primitive features retained from amphibians. So the first reptiles were transitional forms, partway between the amphibians and the later groups of reptiles, just as things like Anthracosaur amphibians were transitional forms. They had some reptilian traits, but not enough to call them reptiles. And so here in red, there are a number of features, including a notochord, which still ran the length of the body, which show that these amphibians were in fact primitive. There weren't that many changes which had to happen, say in the skull. If you were to go from one of the earliest amphibians to one of the earliest reptiles, tens of billions of years had passed, and the skull of the earliest reptiles made of essentially all of the same bones that one would find in these amphibians. And as I'll point to in just a little bit, the earliest reptiles did not have any openings for jaw muscles in this region. So they were missing openings, and there's a name for that condition known as being anapsid. But this then brings us to the features of the group, because while reptiles are certainly important today, the first reptiles were not only the ancestors of, say, lizards and snakes and crocodiles, but also of birds, and then also of mammals, including humans. So mammals, birds, and reptiles are what we refer to as amniotes. Therefore, the very first reptiles, the very first amniotes, are significant because they evolved features which has not only led to the success of modern reptiles, but also then modern birds and mammals. One of the obvious things that one could focus on is reproduction. So here you see some toads. Now, toads are terrestrial frogs. I mean, they have a drier skin. They're found in more terrestrial habitats. But they reproduce in water, as you saw there. Red Fs, that's a larval form of eastern newts. They're very, adept at living in dry terrestrial environments. But here you see the adults mating in water and laying their eggs in water. So amphibians having this, quote, double life ties them to water. Here, in contrast, you see water snakes. Water snakes are reptiles. Now, obviously, they reproduce in water. My fault. Obviously, they live in water, hence the term water snake. But notice them mating. They are mating on land and in a different way. So while amphibians often release sperm and ova together, and fertilization often, especially in most frogs, occurs outside the body of the female, these water snakes are mating. And the fertilization is occurring inside the body of the female. And so the penis in males, and in most groups, the clitoris in females, this is something that only amniotes possess. So the amniotes share features. These features apparently evolved in the earliest amniotes, the reptiles going back to the early Carboniferous period, and are shared by their descendants today, the mammals, the birds, and the modern groups of reptiles. And so thus, these first amniotes were significant, not just in the history of life. Birds, reptiles, and mammals have obviously, played major roles in the diversity of life on land and even in many aquatic environments. But if we were to consider ourselves and how was, say, the human body plan developed, since we are amniotes, some of these amniote features evolved in the earliest amniotes. Now, as I go through these videos, obviously each of the videos can dwell on these in greater detail. I'll mention a few more features. Zooming and playing videos slows them down, and so obviously these videos will be a bit more continuous if you wanted to watch them, on your own individual. Okay, and so amniotes form this biological group, which evolved from amphibians, which have become more and more adapted to life on land. Let me just go back to reproduction. These are named for an amnion, which gets back to that aspect of reproduction. So one of the most significant aspects of this female painted turtle will spend most of her life in the water. But nevertheless, look at what she's doing when it's time to reproduce. She's coming out and she's laying her eggs on land. So the reproduction of amniotes is not tied to water in the same way it was in the amphibians. Amphibians, which often have fertilization occur in the water, whose young develop gills. So if you were to look at larval amphibians, larval salamanders have external gills. Tadpoles have external gills for the first few days and then internal gills. And so here, amphibians are tied to water for reproduction. And that is just inconvenient in many parts of the world. In many parts of the world, it is arid. The rains may only come at a certain time of year. And so while amphibians are better adapted to life on land than fish were, nevertheless, this is one limitation when it comes to reproduction. They are still then tied to water. Here you can see tadpoles in the first couple of days, and you can see their prominent external gills. So what amniotes developed in essence were a series of extra-embryonic membranes so that they could do this, so that they could lay their eggs on land without having to develop an aquatic larval stage. If you were to look at this cat embryo, look at this membrane. This membrane is made by the embryo. These are the embryo's cells. But these embryonic cells are not forming part of the embryo's body, but rather they are wrapping the body in a membrane. And so embryos do that, where they wrap themselves in a series of membranes, which are made of their own cells, but are not forming part of the body proper. So amniotes develop these 4 extra embryonic membranes, which then allow them to reproduce on land. The yolk sac, the yolk is enclosed in a membrane. So, yolk goes back to, the amphibians as being the food source for embryonic development. But then 3 extra embryonic membranes are unique then to the amniotes. The first is called the amnion. It surrounds the embryo in fluid, an amniotic fluid here in blue. So there is a membrane, an amniotic membrane, and thus the embryos of amniotes, even if they're developing on land, are nevertheless surrounded by water during their early development. So these amniotes did not have to seek out a pond in order to reproduce. Their young, their larval forms can develop in a fluid surrounded by this membrane. So birds, mammals, and reptiles are referred to as amniotes. They all share this feature of the amnion. They also share a chorion. That's the extra-embryonic membrane depicted in red here. This performs gas exchange with the outside world perhaps through an egg shell. But then in those which bear live birth, whether it be some live-bearing reptiles or live-bearing mammals, this chorion in red can perform gas exchange with the body of the mother in mammals, forming the fetal part of the placenta. Also an amniote feature in green is the allantois, an extra-embryonic membrane which collects urinary waste so that they are not fouling the embryo as it develops. So because of these bags, these extra-embryonic membranes made of the embryo's cells but not forming part of its body proper, the amniotes can develop inside an egg. They can develop on land. And so the amniotes then no longer tied their reproduction to life on land. So that was certainly significant, and some of the anatomical features which unite the amniotes are reproductive features. Once again, amniotes are the only organisms which possess, the penis, the clitoris, and other reproductive modifications. But there were also many other modifications in other systems, like the respiratory system. This salamander has to breathe by drawing in air and expelling it using throat muscles. That's not very efficient. It just doesn't work very well. And as a result, amphibian lungs, they're not that good. Most salamanders in the Northeast United States don't even have lungs anymore. This is not a good way to breathe. In contrast, the amniotes, they determined that if you can just change the volume of the thorax using the muscles between the ribs, this would be far more efficient than trying to draw air in through these throat muscles. And so amniotes develop longer ribs, which contact the sternum. And so now when the muscles between the ribs contract, I make my chest bigger, but then air comes in at the same time. And so rather than draw it in through contractions of my throat muscles, if I do this and open my mouth, air goes in for free. And then when you change the volume once again, the air leaves. This is a far better way of breathing. Mammals would add to that by adding a diaphragm, which pushes down on the guts and then can make the lungs longer with each breath. And so the amniotes, first the reptiles and then mammals with later modifications, developed better ways of breathing. And this then allowed amniotes to get bigger than amphibians can, to move faster, to be able to exchange more oxygen and support active metabolisms, including being warm-blooded. And so this was a great advance for the amniotes. And it had repercussions that one might not automatically think of. In amphibians, the breathing mechanisms, the lungs and this breathing in oxygen through the throat, it works so poorly that many amphibians have to, depend on breathing through their skin. And as I had mentioned, there are salamanders which do all of their breathing through the skin because, their form of respiration is so inefficient. But once reptiles developed a better way of breathing, notice, this lizard's ribs continue to move air, then the skin was no longer needed to breathe. And therefore, the skin could then be modified. It could develop dry, hard scales, which then in reptiles allows them to live in hotter, more arid environments where amphibians can't live. Later descendants of the first amniotes would then modify scales to become feathers. Mammals would evolve hair. None of these would be possible if the skin needed to be thin and moist to breathe through. And so the improvements of respiration in amniotes then also allowed for the evolution of scales, feathers, and hair. So respiratory changes unite the amniotes. So too did skeletal changes. One very important skeletal change was the strengthening of the vertebral column. Now, one might think that the vertebral column is so complex that it kind of would have had to have appeared in its complex state But that is, the farthest thing from the truth. The vertebral column, in the sense that we think of it, it took hundreds of millions of years to evolve. Before there was a vertebral column, there was a notochord. This is the stiff rod that existed in the back of those primitive chordates and the earliest fish. After then the notochord, fish such as lampreys and early sharks then developed little pieces of cartilage over the notochord. The notochord was still the primary support rod, but now there was cartilage in addition to it, which could anchor muscles. Later fish then also developed cartilage under the notochord, but the notochord was still the primary support rod, and now simply had cartilage above and beneath it. Then as fish lineages evolved, some of them strengthened this by making the cartilage larger and more prominent, and then even fusing the upper portions to the lower portions. Now, it should be noted these fused around the notochord. They did not replace the notochord. The notochord was still a continuous rod that went through this. And when the first sarcopterygian fish came onto land as early amphibians, this was the condition where the notochord ran through these elements which surrounded them. Now, reptiles would make the blue elements smaller and smaller. So the elements underneath the notochord, there were two different types. There were intercentra and pleurocentra. And reptiles reduced the importance of one of the sets. And then the other set then fuses with the neural arches above to form what's called a composite vertebrae. So when we think of a vertebra, say a thoracic vertebra, it seems to be one solid piece of bone, but it evolved by multiple structures which then fused to form a composite bone. Even in human embryos, vertebrae form as multiple pieces of cartilage which gradually surround the notochord and then replace it. And so whether it be in the evolution of vertebrates on land, or even we can watch this in human embryos, we see this process. So the early amphibians had cartilage becoming bone, which was around the notochord. The early amphibians still had a continuous notochord. This was even present in the first anapsid reptiles, but then these elements then fused to squeeze out the notochord, so that the notochord was no longer a continuous structure. So these are features which amniotes share. And as I had mentioned, even human embryos retain some of these features. We still retain a yolk sac and an allantois, even though, given that we develop inside our mothers, we do not need them for their ancestral roles. Those vertebrae still form in the same way that they had in more primitive animals. And so one can see connections to the ancestry of these traits as we study fetal development, including that of humans. There are so many other traits which we could mention. I just want to zip through them quickly. If we were to take a skull of an amphibian in the Devonian period, there weren't that many modifications which would be need to be made to convert this into the skull of a carboniferous reptile. And so all the same bones were still present. You even had primitive features such as those prominent teeth on the roof of the mouth and this pineal foramen here between the parietal bones. We see the connections between these amphibians and the reptiles in all throughout their skeleton, whether it be the vertebral column, the skulls, the hips and shoulder bones of the first amniotes were just modified versions of the early tetrapods, ancestral amphibians, etc. We can point to muscular changes, etc. And so the amniotes are a group of tetrapods. From the ancestral tetrapods, the first amphibians, then there was a subgroup which evolved from amphibians which better adapted to life on land. And then the amniotes, which evolved from them, the birds, mammals, and reptiles, thus share features that modern amphibians lack. So they are a real biologic group, and we can see that in the skeletal features, the reproductive features, etc. Now, to talk about the first reptiles then of the early Carboniferous period, it's confusing because one, they were very small, as I pointed out in those earlier images. And because some of the features which I used to be defining elements of the reptiles, such as reproduction on land, extra-embryonic membranes, even things like scales, none of these things would fossilize. The first reptiles are very similar to the skeletons of ancestral amphibians. The most important features from the fossil record. Then how does one decide what is the difference between an amphibian, which is almost a reptile, and a reptile, which is very similar to ancestral amphibians? And the answer is it's difficult, and there are fossils which experts could argue about. How should one classify these? Are these advanced amphibians, which are very close to being reptiles? Are they very primitive reptiles, very close to being amphibians? Should we use the term para-reptiles to include animals which have some but not all of the features of reptiles, etc.? And so we could, once again argue about where to put many of these. Now, all the ones given here occur after the first reptiles, and so none of these would be ancestral. But once again, it's significant that reptilian features evolved in stages. And here we have a group which we could argue, have some of the features, but not all, and perhaps, not enough to truly call them reptiles, but we need a group of amphibians, which is a sister group of reptiles. The very first reptiles of the Carboniferous period were small insect eaters, most likely, give or take a foot long. And I showed you some of their fossils earlier. And so while reptiles would later be far more prominent, they have very humble beginnings, which was probably necessary. If one were to now evolve mechanisms to reproduce on land, they no doubt would not be fully functional at first. And so therefore it makes sense that, perhaps the earliest reptiles to reproduce on land were saying, laying eggs in, ponds which were drying up or under moist logs. And this might work if they were small, and this development took place, in a short period of time. So had they been larger, that probably would not have afforded, the evolution of, these new membranes, because, simply the embryos wouldn't have been able to, support themselves, or, a moist environment might not have stayed moist for that entire time. And so in the Paleozoic era, we have a number of these small reptiles, and it was probably in these groups that some of these, mechanisms were improving and becoming more important. Now, I'll be talking about the holes in the heads presently. Milleretta is significant for turtles in that it started to have thickening of ribs, as I'll get to presently. Okay, so the earliest reptiles were what were called anapsids, and I'll describe what I mean by that in just a second. And because there are no lizards yet, even though these animals might look lizard-like, they are not lizards. There are no crocodiles, no snakes, no turtles. There are no dinosaurs. That there was significant variation in this group. Now, I'll be talking about turtles later, but I'd like to just introduce some of them now. Some of these reptiles then evolved thickening ribs, all right? Milleretta had thickened ribs, and not just thicker ribs, also showing some similarities to the ribs of turtles. The same it was true of Eunotosaurus, where some studies have included this in the turtle clade, not as a turtle, but as like a sister group of turtles. So among these anapsid reptiles, there were some features which are important for later groups. Also wanted to point out, here's one which was, approaching 20 feet in length. Reptiles began to eat plants. Amphibians really don't. I mean, even though tadpoles are largely herbivorous, depending on plankton and algae. Adult amphibians today are all essentially carnivores. And so it was the reptiles that then began to adapt in many lineages to eating plants. If you eat the green stuff, then you can get to be bigger. All right, there are certainly large herbivores alive today, giraffes, elephants, cows, moose, etc. And so reptiles were the first, terrestrial tetrapods to really adapt to eating life on land. Now, as I had said, these are anapsid reptiles. But then let me go back to what I mean by that. When we chew our food, there are muscles which attach to the lower jaw that then attach to the bones of the upper portions of the skull, to allow this chewing of food and allow us to bite and to close our jaws. If, however, these muscles were limited so that they ended here, smaller muscles would mean much less bite force, and we wouldn't be efficient at biting. However, our jaw muscles, they go through a hole. So here we have an opening in the vicinity of the temporal bone. Here we have a zygomatic arch. And so on the other side of this arch, these muscles can actually go through a hole. And then thus the muscles attaching to our jaw do not have to be as short. They can be longer, which thus gives us a stronger bite. The first reptiles did not have that hole. So in Latin, they were anapsids. They had no opening in the temporal region of the skull, which then probably limited, their feeding and the bite force which they had. Later groups of reptiles would develop holes, and we would then, use potentially the presence of a hole to name one group as the synapsids, and then another group as the diapsids if they had two holes. Now, that distinction, you either have no holes and you're an anapsid, you have one hole and you're a synapsid, you have two holes and you're a diapsid. That's nice. Very often when we use anatomical traits, anatomical traits may not be perfect. So for example, tetrapods, they have 4 legs. Okay, except for snakes, which don't, and caecilians which don't, and whales have lost their hind legs, etc. And so sometimes there's a really good feature like the presence of limbs, which in general is good for classifying a group, but which there are these exceptions that one has to acknowledge. Temporal openings are a good feature for breaking the amniotes into groups, perhaps not a perfect one. So as I'll get to later, the first reptiles have no temporal opening. The first turtles have no temporal opening. And so anatomically, one would argue that turtles would then be modified anapsids. Genetic studies suggest otherwise. Genetic studies lead to inclusion of the turtles in the reptilian groups which have openings. So what does that mean? Does that mean that, say, after mass extinctions, the rate of evolutionary change was so modified that maybe the genetic analyses, aren't as reliable for the specific, branchings as they might otherwise be? Might it be that here was an anatomical feature, these spaces in the skull, which were later lost, just as some groups lost limbs which once had them, did some groups lose temporal openings which once had them? And so, as I'll mention later, there has been for quite some time disagreement on how to classify turtles, and this temporal opening presence or absence might lead one to a different conclusion than the genetic analysis. That's perhaps a little further afield. So the first reptiles were anapsid reptiles. From these anapsid reptiles, the synapsids would evolve. Synapsids had one temporal opening in this region of the skull for jaw muscles. And the synapsid reptiles would do wonderfully in the remainder of the Paleozoic era, evolving into pelycosaurs, therapsids, and then even cynodonts. Synapsid reptiles have since become extinct, but mammals are synapsids. So if you look at this human skull or this monkey skull or this, I'm sorry, I think that's an opossum skull, you can see this prominent synapsid opening. So that synapsids are derived from the first reptiles, which were anapsids, and they include not only the mammals of today, but the synapsid reptiles, which were dominant in the Paleozoic era and are sometimes called the mammal-like reptiles. Most reptiles in history were diapsid reptiles, which had two temporal openings. So here on this crocodile, notice that in the temporal region of the skull, there are two openings which allow the jaw muscles to then become longer and attach here. And so the diapsids would include dinosaurs and the birds which evolved from them, but then crocodiles, lizards, snakes, and a host of reptiles in the fossil the fossil era. So in future videos, I'll obviously be going through the diversity of the diapsids, which would include lizards, snakes, dinosaurs, birds, and then also the synapsids, which include mammal-like reptiles and mammals. But for right now, I would like to then just return to these anapsid reptiles. They are the very first reptiles. The first reptiles are anapsid reptiles, and they virtually all become extinct at the end of the Permian. So once again, the amphibians, which lack any temporal opening, evolved into anapsid reptiles, which lack any temporal opening. But from anapsid reptiles, there then come these two groups. One group has one temporal opening and is a synapsid. One group has two temporal openings and is a diapsid. Okay. So once again, it was amphibians from the Devonian, which evolved into reptiles by the Carboniferous period. So the very first reptiles in the early Carboniferous are anapsid reptiles. By the late Carboniferous, there are both synapsids and diapsid reptiles. Now notice that the Paleozoic era is about to end. It ends in the worst mass extinction in Earth's history. And so as we consider the end of the Paleozoic era with these, say, Parasaurs, which I'll get back to presently, these are anapsid reptiles, and then they die at the end of the Permian period when the volcanic eruptions in the Siberian traps were affecting the Earth, as was the final effects of the fusion of the continents. So this certainly has a large impact on life on Earth. More than 90% of life dies out. And so many of the synapsid reptiles that you see here, these are the quote mammal-like reptiles, they become extinct in this mass extinction. And so while synapsids were dominant at the end of the Paleozoic, they would not be the dominant group of reptiles in the Mesozoic era. But then getting back then just to the last of the Anapsids. So I'd like to just show you a fossil skull or two, and you can note once again that there is the absence of those holes here. So you see the human skull. Notice this hole allows the jaw muscles to become larger, but the early reptiles lacked these. They were anapsids. And so, for example, here is a skull of a primitive reptile. Notice the absence of an opening in the temporal region. Now, some of these would actually become quite large, as I had mentioned, that they started to eat plants. So paraeosaurs were this, a dominant group of plant-eating reptiles among the largest animals on Earth. And look at how big this skull is sitting on this chair. So reptiles were able to adapt, to terrestrial environments in ways that amphibians had not been able to. So some thickened their ribs. That's important for reptiles, for turtles, which I'll mention presently. And here I just wanted to point out these paraeosaurs. They were huge anapsid herbivores. But while anapsids did achieve some diversity and some success in the end of the Paleozoic era, the ending of the Paleozoic in that mass extinction would doom many lineages, including these paraeosaurs, many of which had armor in their skin, dermal bone, which then thickened their hides. Some, then had extensions from their skulls, which would form horn-like defensive mechanisms, and so they were a major group, but they became extinct. But then that gets us to the question of turtles. And once again, there has been, so here's just some of the diversity among these paraeosaurs in their skulls. So turtles did not begin with a temporal opening. So they had the anapsid condition. Now, we could, postulate that perhaps that was because one group of diapsids lost these temporal openings, modified, the skull, once again. So perhaps the absence was a secondary loss. Perhaps, some of the changes in these first reptiles, are still being, elucidated. And once again, so there is disagreement as to whether, these turtles hail from an anapsid or a very early modified diapsid lineage. There are anapsids which seem to be similar to turtle in having thicker ribs, Milleretta and Eunotosaurus. And so before there were turtles, there were some Permian reptiles which had turtle-like traits. After the mass extinction in the Triassic period, there were actually then turtles. Now, Odontochelys had thick ribs and had dermal bone on its underside, forming a plastron, but did not yet have a carapace, which, a dorsal part of the shell. It was clearly not any modern turtle because, it still had features like teeth on the sides of its jaws and teeth on the roof of its mouth. Later turtles, such as Progonochelys, also from the Triassic period would lose the marginal teeth, but would retain the teeth on the roof of the mouth. So from primitive forms like Odontochelys, which didn't even have a complete shell, there are other primitive turtles known such as Proganochelys, which now have a complete shell. I do not believe that the two halves were fused as in modern turtles. And they were also unlike the shells of modern turtles. I mean, there were these extra bones here. There was this extra row of bones in the shell that no modern turtle has. And they still had teeth on the roof of the mouth. So the turtles of the Triassic period were not modern turtles by any means. By the Jurassic period, there are turtles which are classified as pleurodires, and the ancestors of the cryptodires, which are the major group of turtles which then can retract their necks, although retracting necks probably did not appear until the Cretaceous period. So just some final points in wrapping up turtles. One is that there certainly are reptiles which have some but not all of the features of turtles, whether it be thickened ribs. Other lineages had dermal bone in the skin, and so modern turtles certainly do not appear from nowhere. The first turtles are transitional forms, which Odontochelys had only part of its shell. Proganochelys transitional with many intermediate features. From these early turtles, which belong to no modern groups, we get the turtles of today. So we classify turtles together in a group because they share features like that shell. But here you can see a pleurodire turtle. It is a long-necked turtle which is not capable of retracting its neck inside its shell. So going back to the Jurassic period, there were pleurodires which had this feature. They had shell, but they wouldn't allow retracting inside the shell. And today, the pleurodires are known from a number of the southern continents. So remember that at that period, the continents were to separate, and the southern continents formed the entire throughout some of the Mesozoic or turtles which are today, once again, South America and also in the southern continents. The second group, the cryptodires, of retracting their necks inside their shells, and these then evolved into separate families, which we then have today. And I have a few videos here which go through different families of turtles and where you can find them today. Different ones adapted to life on land, and so this is from various which are simply turtles that are adapted, as I said, and through just so the turtles evolved in series of the follow shows for turtles. Too, all right, and ribs and having dermal bones associate to form. This is not control used to dermal scales, scutes, which attach then to it. Also of interest is that turtles migrated their turtle to the inside of the which is so as I had mentioned, there are a number of things which made turtles distinctive. In addition to the shell, they also then modified their shoulder girdle so that it was actually inside the rib cage as opposed to the typical outside the rib cage. Now, how could that have been managed? Well, once again, if one were to look at Unitosaurus and Milleretta, one sees some transitional forms where some turtle-like features. But both from studying the first fossil turtles and then turtle embryos, one can see that the ribs in, so here you can see the shoulder girdle is actually inside the rib cage. The way that that was managed was a transitional form where these ribs then just became shorter. And so there was a brief period where the first ribs were shorter in structure, which allowed then a movement of the shoulder bones and the muscles into this space now occupied by them. And so once this rib became shorter, all right, then here you see some of the muscles, which then allowed for this shoulder girdle to be moved inside this rib cage. And this process is seen in turtle embryos. And the first turtle fossils have short ribs here. So this video has, been perhaps a long introduction into the amniotes. Between the end of the Devonian period, when there were tetrapods living on land, and the early Carboniferous, a new group of vertebrates evolved on land, which was adapted to life on land. They could reproduce on land. They evolved better ways of breathing and supporting their weight on land. These became the first reptiles, the first amniotes, which is significant given that reptiles, birds, and mammals today are amniotes. Those Carboniferous amniotes were the ancestors of dinosaurs, mammals, birds, and the most successful groups on land. The very first amniotes were Anapsids, which did diversify a bit, but which became extinct at the end of the Permian in that mass extinction, with the possible exception of turtles. So anatomically, one could make the case that turtles were a surviving lineage of Anapsids. But genetic evidence seems to suggest that an early group of diapsids was then modified, and the turtles derived from that lineage.